Uh, Today, I'll be talking about my work in collaboration with the Borovets Laboratory at Colorado State University to infer the phylogeny of the wasp family Trigonalidae. Most trigonalids are hyperparasitoids, which is to say that they are parasitoids of other parasitoids, but regardless of whether that's the case or not, all trigonalids have an ultimate host, a parasitoid fly or wasp, or sometimes a social wasp, which they reach by way of an intermediate host, a caterpillar or sawfly larva that swallows trigonalid eggs laid in foliage, as seen in this picture. Only if the intermediate host is then attacked by an ultimate host, attacked and eaten, do those eggs hatch and develop to maturity. So, given the convoluted series of events that are required for a trigonalid wasp to develop, as you can imagine, they are extremely rare and have seen very little representation in molecular phylogenies of the Hymenoptera as a whole, that being the larger group to which they belong. And in fact, to date, there have been no molecular phylogenies focusing on the Trigonalidae. The only phylogeny whatsoever to focus on the Trigonalidae was inferred from just 30 discrete morphological characters and used maximum parsimony. But unfortunately, it appears that morphological evolution in the Trigonalidae is anything but parsimonious, regardless of the phylogenetic hypothesis in question or where one roots that phylogeny. It's clear that multiple characters have been subject to secondary reversions from a derived state. So any phylogeny inferred from morphology alone is suspect. For a moment, I'd like to focus on the putative clade Nomadinae, inferred from morphology alone and shown here in gray. Most nomadinines are primary parasitoids of Vespidae, that is, hornets and their relatives. It must be emphasized that this evolved secondarily from hyperparasitoidism, which seems to be the ancestral condition for the Trigonalidae. Some nomadinines are instead facultative primary parasitoids of solitary vespids, such as this otter wasp shown here, but most are instead parasitoids of social vespids in the Polystinae or Vespinae, with an example being shown here. Among these, the genus Nomadina is particularly interesting in that they lack palps around their mouth. For comparison, this is what the palps usually look like in the Trigonalidae. What this means mechanically is that Nomadina cannot feed themselves, and although we have never or almost never directly observed them alive, this suggests that in addition to being primary parasitoids of Palestine wasps in this case, they are social parasites that integrate themselves into the host superorganism and beg for food, analogously to the parasitoid wasp shown here, which attacks ants. To understand the evolution of behaviors like this in the Trigonalidae, we need to infer their phylogeny, which we did from 122 ultra-conserved elements with taxon representation of 13 out of 15 described genera. We inferred a species tree under a maximum likelihood framework in IQ tree, and also a species tree using a two-step coalescent-based approach, with trees inferred for each locus in IQ tree and used as input into Astral. This accommodates the effect of gene tree discordance. And we recapitulated these analyses with a subset of 89 ultra-conserved elements, excluding those that did not conform to the assumptions of compositional homogeneity and stationarity. The hope here being that inference from these data alone would be less subject to systematic error, at least with respect to those particular qualities inherent in the data. So for our maximum likelihood tree inferred from the full data set, we have four major, very well-supported clades, which incidentally do not correspond whatsoever to the existing subfamilial classification, but ultrafast bootstrap support for the relationships among them is quite poor. The same analysis with a different data set recovers the exact same clades, but the relationships among them have been completely rearranged, and again, very poor ultra-fast bootstrap support. And with coalescent-based inference, we get a third view of the relationships among these four clades, which is consistent irrespective of data set, but again, support for the relationships among them is quite low. You'll note that the local posterior probability for those relationships is reduced or diminished in coalescent-based inference from the reduced data set compared to the full data set, which to me implies that the relatively elevated local posterior probability in this phylogeny is an artifact of model misfit, perhaps. I also tried to visualize phylogenetic signal in our data using four-cluster likelihood mapping. That is the signal with respect to the relationships among these four well-supported clades within the trigonality. And you can see that there is a great deal of conflicting signal in the data. In fact, when we look just at the reduced data set, signal is pretty much equivocal for these three different hypotheses. So, in the end, so far, we have not yet resolved relationships among the four major clades of the trigonality due to a great deal of discordant phylogenetic signal. So resolving that will be the main task for understanding the evolution of the trigonality. From a classificatory perspective, one of these possible weakly supported hypotheses is appealing because it does divide the trigonality into two clades that are diagnosable based upon morphology, which I'm informally calling crunchy boys and soft boys. These are distinguished by differences in their tergosternal thickness, and also quite consistently by the shape of the propodial foramen. But the most important conclusion from our results so far, in my opinion, is that the putative clade Nomadinini is in fact polyphyletic. And the implication of this is that primary parasitoidism, or obligate primary parasitoidism of the Vespidae in the trigonality has evolved at least two times, as seen in this phylogeny. I say at least because if we focus on the clade that I'm referring to here as the Seminotini, the statistical support for relationships among the lineages within this clade is always weak, but in every phylogeny that we inferred, there are two separate origins of obligate primary parasitoidism of social wasps. The remainder of the members of the Seminotini are facultative hyperparasitoids of solitary wasps, like the potter wasps that I mentioned earlier. Moreover, it's worth noting that four-cluster likelihood mapping of these relationships does indicate favor in the alignment for one origin. So again, a case of a lot of conflicting phylogenetic signal. However, even just two origins of obligate primary parasitoidism in the trigonality is notable because this shift away from hyperparasitoidism is unique among lineages of hyperparasitoid insects, so far as I'm aware so far. So, we have a lot of future research objectives for this project, all of which center around Bayesian total evidence inference of both divergence states in the trigonality and ancestral state reconstruction of the biogeographical history of this clade. We intend to perform this under the fossilized birth-death model. There are only two definitive fossil representatives of the Trigonalidae, which is unsurprising considering how rare they are. But thankfully, the Trigonalidae are sister to an extinct clade called the Mimetidae, which do have a pretty rich fossil record, which can inform, then, the fossilized birth-death model under which we're trying to infer the divergence times within the Trigonalidae. With this, we hope to illuminate the timing of biogeographical shifts in the Trigonalidae. They have actually a pretty convoluted biogeography on the whole. I think that the genus Orthogonalis is a particularly interesting example that I want to understand the history of a bit better. They definitely appear to have originated in East Asia. There is a clade in the Americas. And this relatively depauperate clade within the Americas is definitively sister to a more species-rich clade within the Afrotropics and into Madagascar. To my mind, or to my knowledge, this is a pretty unusual biogeographical distribution, and I'd be interested to hear from anyone if they've seen anything like it in the past. And with that, are there any questions?
Hey, thanks our speaker, and yeah, I think I see one. Is there one hands up?
Yeah, that's me. Lovely talk, very good stuff. I had a question for you about sort of homoplasy in the morphological traits, because this is an issue in ants as well. And so do you think that lack of resolution is due to the fact that, there's a large amount of species and relatively small number of characters, or do you think it's just there are only so many ways to be a wasp? And sort of once you get into that shape, your morphology is constrained.
With trigonalids, it's not because there are too many species. They are quite species poor as far as insect clades go. Um, they really are very labile morphologically, and this is something that I chose not to address in the talk in the interest of time, but there's, I think, a rather absurd amount of convergent secondary reversals of putatively derived characters, and as I said, um, that is the case regardless of how you root the tree. So for trigonalids, at least, I think they are, it's not just that that they show a lot of homoplasy, because that's something that we see a lot. And as you alluded to, we certainly see that to an extreme degree in ants. It's that they're also flip-flopping back and forth in a way that is, as I said, not parsimonious. But we can't really understand what this looks like until, of course, we have a robust phylogeny.
And so do you think that lability is something about the group, or do you think it's just the type of traits that people tend to collect in this group?
There's only so many traits that can be collected. I don't think it's acquisition bias. I think it really is the group itself. And I think it has perhaps something to do with the fact that they're hyperparasitoids and their body size is actually quite variable on intra-specific level, and not just body size, but also coloration and even antennomere count varies wildly within species. And I suspect that maybe that somehow gets extrapolated on a macroevolutionary level, but who knows?
Yeah, thanks. I think we're going to move to the second speaker. Let's welcome Brooke Long-Fox as our second speaker.
Hi, thank you. Let me just figure out how to. Sorry, trying to get to presentation mode. There we go. Okay, can everyone see the screen? Okay, perfect. Thank you, everyone, for having me here. So today I'm going to talk about reproducible morphological data for phylogenetics, and there's some new developments for MorphoBank. So what is MorphoBank? MorphoBank is a data repository for morphological character data. It stores images, metadata. You can make projects with different character matrices. You can make projects with just images of different specimens and taxa, and everything is linked with a DOI. So there's DOI for the matrix. There's a DOI for different folios of images. This helps make the data reproducible. So a little bit about, morphological character matrix. For different phylogenetic trees, you may have things like yes or no. I'm gonna quickly skip through this so I don't take too much time. But for example, for your matrix, you could have something like characters that are, say, character tail, elongated or short, zeros or ones. So the matrix is composed of zeros and ones. So what MorphoBank is it's a repository just for this type of data. And so it's majority matrix data, but we do also allow images. And so why for your project or your research, why would you put your data into MorphoBank? And it's partly because of reproducibility. So we want our science to be reproducible. Some common issues are that matrices are frequently unpublished. Maybe they become unavailable after peer review. Different images for your data and your different specimens could be scattered across different supplementary files, or they're absent altogether. Characters are often poorly documented, so it makes reproducibility of scoring. So if you decide to put something as a 0 or 1, it makes it harder to evaluate for peer review or for someone trying to replicate your data or to build onto your matrix or to build onto your tree. And then data, usually when you publish your research, a lot of times this data goes missing. So your tree gets published, but the data to build that tree is gone. And so when a new person comes in to do research, maybe they want to add a, a specimen or a different species to a tree, you shouldn't have to reconstruct that data. It would be great if you can build upon it. So kind of like how we have GenBank for molecular data, MorphoBank is set for morphological data. This data can be used in many different kinds of analyses. So it's not just for phylogenetic analyses. It's used in macroevolutionary studies, comparative morphology, and conservation paleobiology. So why would you benefit from sharing your data? Some of the author benefits include more citations. So if you share your data, the data set itself can be cited. So it's not just your published paper, but the data can be cited and reused. Sharing data also makes your work more visible. So it becomes discovered, it can be reused. It also adds to transparent research. So reviewers, journals, editors are increasingly expecting your data to become accessible. And so whether you deposit, your morphological data in MorphoBank or you deposit your data somewhere else, this is a common thing that should be continuously done. And then reusability. So once you publish your research, if you have your data stored somewhere in a public or open access repository, that's great because then your data doesn't get lost after publication. You don't have to worry about someone emailing you down the road and saying, hey, can you share your data from this paper? You could just point them to the DOI in the repository. And then there's also collaboration opportunities. So when you share your data, other people can see it. They can build off their work, they can contact you, and then you can be part of more research projects for that. And then you also get credit. Most repositories, when you deposit your data, you get a DOI, you get an ORCID link. So now data is also counted as a research product. So instead of just having the paper be a product, the data is a product as well. And so for MorphoBank and a lot of other repositories, when you publish your data, you get that DOI and you get that updated and linked to your ORCID. So it's easier to find the data. There's also, besides just personal benefits, there's open science benefits. So publishing your data means that there's no paywalls and anyone can view and use the data regardless of institutional access. This is great because it supports equitable science. Researchers from underfunded institutions and countries can fully participate. There's no barriers. In addition, that it benefits education and outreach. So when you publish your data, it's not just researchers that can see it. Teachers and students can download your data and use it, and they can actually get their hands on real data. So this can be great for classrooms, for K through 12 education, for any of your undergrad or graduate classes you may be teaching. You can use this data. Okay, so a little bit about why repositories matter. So emailing a file to a colleague is not necessarily data sharing. The repository makes data findable, usable, and permanent. This helps with data preservation. So data are stored long-term. They're independent of author career changes, or if you move to a different institution, the data is still there if it's in a repository. Most repositories require metadata. So this is the structured context of what makes the data interpretable by someone who wasn't in the room when it was created. You also get searchability. So the data set itself can be discovered and reused by researchers who didn't know that they existed. You get a citation. So DOIs give data sets more formal credit. You can also link them permanently to an author through things like ORCID. And then there's peer community. So repositories can connect your data to the broader scientific community. And this helps build reuse. It helps build community trust. So the role of specialized repositories. So like I mentioned, I'm here to kind of share a bit about MorphoBank and some of our updates, but there's a lot of kinds of repositories. So there's general repositories, and those are usually just file storages. So Dryad, Figshares, and Nodo, these things allow you to just take your data and upload it. And so it's really just a file storage system, and it really lacks discipline-specific structure. So multiple people could be working with the same type of data. If they upload it to one of these general repositories, it could be in a different format or various different formats. So it makes reusability a little bit difficult. Then there's specialized repositories. These are repositories that require a structured format. For example, MorphoBank is one of these structured and specialized repositories. And it takes a bit more time to get your data in a specific structure, but it helps make the data interoperable because there's standard formats for MorphoBank. This would be like a Nexus or a TNT file. And then the structured format also makes the data reusable. So this helps maintain relevant metadata and structured data organization. And then in specialized repositories, you often have curators or staff that are subject matter experts, and so they can help you format and edit your data. So I already kind of went over what is MorphoBank. MorphoBank helps make data fair. Fair means findable, accessible, interoperable, and reusable. Findable is the DOI. So it can be located, it can be cited. Accessible means that the data and metadata are standardized. And this means that even if for some reason, if the data were to be corrupted, the metadata still persists and has some information associated with it. Interoperable is a standard format. For example, with phylogenetic software, this could be a Nexus file or TNT, and it makes data reusable. So data are usually licensed. You can pick what you want if you want your data to be reusable. So that way people can legally build upon it. So what kind of data is in MorphoBank? Right now we have over 1,000 phylogenetic matrices. We have a global user community, which represents hundreds of journals. Everything in MorphoBank is open access, so you can go online and deposit your data or access the data with no paywall. This is just some of the most up-to-date metrics of what we have. Here's a bit of an example of what a project looks like on MorphoBank. So this is one that's in press, set to come out in 2026. And basically every data set we would like to be associated with a peer-reviewed journal article. So when you go to publish your paper, or usually it's before you publish, when you submit, typically you would deposit your data and you would give the editors and the reviewers access to this data. Each project, and by project, that's the link to the data, is given a DOI. And then we also have a connection to the journal article. So you see the data and you can actually click and read the article, and there's that connection between the data and the research paper. For an example, for this particular project, this is an example of what the phylogenetic matrix would look like, or what the matrix would look like in here. So it's set up pretty standard, but we do have the option. So there's like a show numbers. You can also show the character state information. So this is the option for that. And you can even add photos. So if you have a species and you have a specific character and you have a state, you can show what that state looks like in photo form. So like whether it's present, absent, we even have the option to have your image annotated. So you can go in and like point to specific things to describe what you mean. And this helps with reusability and to make things replicable. You know, if you label things, and it helps for future research. And so some of the improvements to MorphoBank, we have MorphoBank's 25 years old, and we've completely overhauled the infrastructure and the media storage. We've also expanded phylogenetic tree building. Before we just had PALP, now we have Mr. Bayes and TNT. We have ORCID integration, so you can connect with ORCID. We have a new AI-aided character extraction. So before you had to upload your data in a very specific format to be added, and now you can kind of upload it in any format, and you can add additional files, and we have an AI program help with that extraction. You can upload CSVs and Excel files now. You can combine different OTUs in the matrix, and we have just a general user improvements. So this is just a screenshot of our ORCID option. You can now connect to ORCID. We have our AI extractor. I know I'm running out of time. And then just a little bit about this. So MorphoBank is free to use, but it's not free to fund. So we run off of support by various societies and institutions around the world. Okay, I apologize for going a little too long.
Yes, maybe one quick question. Is there anybody raising hands? Okay. I'll ask the question. How does this compare to the older like the MO fashion? So I think I used to use tree base. Is there any implementation that's try to import the data from other repository, or how they compare, or how do they coexist?
So we try to add data from various places, and a lot of repositories are without federal funding are are kind of disappearing. And so MorphoBank has this institutional support, and we try to get different universities to help support it. And so, um, as data is disappearing at other places, it's great if you can add your data here. And I know we're kind of out of time, but thank you,
Yeah.
everyone.
Okay. Thanks for your presentation. So I welcome Daniel Schönberger as our next speaker.
Yeah, hey, can you hear me?
Yes.
Great. Yeah, my name is Daniel Schönberger, and with this talk, I want to present you a new species delimitation approach that is fully integrative by combining multiple data types. So, yeah. So if you think about integrative species delimitation, you often have a lot of genomic data, maybe morphology, but hopefully you will also include other lines of evidence going from climate, behavior, maybe symbiont data, or transcriptomics, life history traits, and so on. So combining all of these lines of evidence basically reduces the reliance of a single data set, which might diverge in a different way compared to other line of evidence. So overall, using multiple lines in this integrative approach increases more robust species delimitation. And it might also allow to give or to obtain insights into the processes behind divergence. To give you a really cool example from a recent study about these lemurs here from 2024, where the authors used an integrative species delimitation approach. They first conducted this phylogeny here, and they then looked at candidate sister pair species. They had this iterative decision tree here on the right, where they first looked at genomic evidence, then morphology, climatic data, and behavior. And just to give you one example here, these two candidate species, where they showed first they used an isolation by distance model, which was inconclusive in their result. Then they used other genomic methods to find that that these two species form distinct clusters and are monophyletic. And they found that overlap between these two species is really low. Based on that iterative decision tree, they then confirmed both as separate species while rejecting to synonymize both. So that's a really cool example about iterative integrative species delimitation. Normally, if you think about comparative, or if you think about integrative species delimitation, it's more comparative, meaning that we have multiple approaches, for example, population genetics, maybe a phylogenetic analysis, then an analysis that evaluates gene flow for genomics. We might test for niche divergence, or we might evaluate or assess host specificity. And in the end, after running all of these different analyses, we will come to an overall conclusion, whether our tags are different or how many k-values are in our data set. So from my own experience, this is takes a lot of time, it takes a lot of experience and expertise to run all of these different analyses. So why not put all of these different layers into one single model and run a fully integrated species delimitation approach? And that's basically what we're working on currently. And this approach is called SOM. It's based on a machine learning algorithm that has been introduced for species delimitation by Alexander Pyron here. And it's called self-organizing maps. And now, together with Alex as collaborator and my supervisor, Julian Dupuy, I'm now further developing this approach, and as well as offering you an R package that has tutorials and is easily accessible and really flexible and simple. And we also include new and improved utilities, metrics, and do a lot of empirical and simulation testing to see how it performs. Just to give you a short summary of this really complex or complicated procedure. So what we basically do is we take this normalized multivariate data that we get and reduce it into this two-dimensional SOM grid that we see here. And after training, after this machine learning algorithm trains this map, each of these cells is basically a representation of our original multivariate space, or one part of that basically. So cells close to each other are also close in this multivariate space. In the end, what we can visualize is the distances from one cell to its neighboring cells. So here in yellow or light values, you see low distances, and you can see high distances between each other in these dark colors. And the main point here is that these low distances in yellow represent two areas that are separated really well by this dark distance. So basically two distinct clusters which can be interpreted as species. And in the end, we can also assign these white little dots, these individual samples that we had in the beginning, back to this trained individual map. That's basically done by comparing our individual sample data with these trained prototypes that we get. And by doing that, we can assign it to the closest match that we can get. So what are the main advantages of this approach? The biggest advantage is that we can combine different data layers that are multimodal, so that can be continuous, it can be categorical, binary, or SNP data into a single model. And the data can also be non-linear and complex. So for example, compared to a PCA, which is mostly based on linear data, this one can deal with such data. We also have additional features, such as we can extract what layers are most important and what variables are most important, as well as getting estimates of uncertainty and some kind of admixture-like ancestry coefficients. One other really cool thing is that this method is really robust to missing data. So you can even give it data with 50 or 60% missingness, and it can still perform really well. Currently, we are testing this by extracting and analyzing different empirical data sets that have multiple layers available. So going from salamanders to sea slugs or corals. And just to give you one of my favorite examples are these Polygonia anglewing butterflies here. So these occur in Western North America and are morphologically really similar to each other. And so we basically used this study to extract all of their data in layers. So they used genomic data, as you can see here. And based on their study, they inferred 4 different species. I will come back to that in a second. We also extracted their C1 data, as well as really cool layers of morphology. So here's the first morphological data set, where they had 6 continuous traits, where they analyzed different wing spots and their color. And they also extracted different categorical and binary traits based on length, based on field guides, and we also included that. In addition to these available layers, we also extracted their sampling localities together with elevation as a spatial layer and extracted a comprehensive set of environmental layers. And all of these 6 layers were then put together into this SOM model, this machine learning algorithm, to train our SOM map. So at the top, you can see the result of that. What we basically see is these 3 yellow areas that are well separated by these blue large distances between cells, which again basically indicates 3 different species in our data set. What we can extract then is basically this admixture or structure-like plot here, where each sample represents one column here, and we can then extract these cluster assignment coefficients here on the y-axis. And what we basically see here is 3 different clusters. And you might now compare that to the original study that inferred 4 different species here on the right. So if you compare that to our clusters, you can see that one of the clusters actually groups 2 species together. And there are several reasons why this might be the case. So the first one is that both of these species are probably closely related and only recently diverged. And if you look at the right, you see And one species also has really low sample sizes from around 10 to 12 samples. And it seems like, based on our experience and testing, that this approach actually requires at least 15 to 20 samples to actually capture this multivariate space really well. In addition, our approach is designed to be actually really conservative to avoid oversplitting and to also avoid capturing population structure as different species. So next, we can also extract what layers are most important, and we're still testing different metrics for that. I won't go into detail here, but what you can see is that SNP data have really, really high values, meaning that they are most important in differentiating those clusters. We can do the same looking at specific variables within each layer, getting estimates of what variables are most important in differentiating those clusters. Just to look into one of those, into morphology, here our model suggested that wing spot 15 and 14 and the red color values of this is most important in separating clusters. We also used extensive simulations to test the performance of our approach. So we're using a lot of multi-layer simulations where we simulate SNP data, morphology, climate, and host data for different K values. And we then can use that to infer how different parameters work or how different clustering algorithms work on that, how the impact is of missing data, and so on. But today I want to show you the SNP data layer, where we basically simulate two populations or two different species with 20 individuals each. And then we gradually increase the divergence time between these to see at what point our model infers k equals 1 and what point it shifts to k equals 2. So normally we would expect on the y-axis, you can see the proportion of choosing k equals 2. It goes from k equals 1 in the beginning, where we have really low divergence time, gradually over intermediate models that have both k equals 1 and k equals 2 to a point of k equals 2. And that's actually what we see in a model of no migration, that at around 60,000 generations, we see a shift going from k equals 1. We have a few intermediate models that have a mixture-like or uncertainty to k equals 2. If we compare that to FST, we see that this approximate time of 60,000 generations in our model, where we see this shift, corresponds to FST of around 0.11. As a next step, we also introduce gene flow to see how our model actually handles scenarios of gene flow. And we can see that low migration and moderate migration rates are actually captured really well by many intermediate steps that suggest gene flow, basically. While high migration is actually captured as staying at k equals 1. You can compare that versus FST values, which basically show the same patterns as before, and that around FST 0.11, we see the shift from k equals 1 to k equals 2. In the future, we're going to use or we're going to compare these results and these approaches to commonly use other approaches such as DAPC or maybe structure to see how our approach performs to these other approaches. And with that, I just want to say that you can access this package or this approach already on GitHub. It's still in development, so there will be a lot of changes, hopefully, throughout this year. And hopefully, I also see you for the in-person talk in June, where we have more simulations and empirical examples. With that, I want to thank my supervisor and our collaborator, Alex. I'm happy to take any questions.
Let's see. I don't see any hand raised, so I'll try to ask one question. So, just in your model, do you need to predetermine the weight of different layers, or they will just try to determine them by themselves? Like, do you prioritize like certain layer to be
You
the major?
can specify it yourself based on maybe prior hypothesis or biological knowledge, but usually it's done automatically by basically taking into account how many variables you have and what their variant structures are. So that if you have a SNP layer with like 1,000 SNPs, right, you don't want to have that to overly influence versus like if you have only 5 morphological layers, from 5 morphological variables. So our model takes that into account basically automatically.
Okay, thanks. So thanks, Daniel, again. We'll welcome our next speaker, Ainun Nadhifah.
Can everyone see my screen?
Yes, it's working.
All right. Thank you everyone for joining this presentation. I'm very excited to share part of my dissertation project about the evolution of branching complexity in leafy liverworts and the correlation between this complexity with the environmental factors such as habitat moisture and substrate preferences. So, what are liverworts? So why do we care about liverworts? So liverworts are non-vascular plants that belong to bryophyte lineage together with mosses and hornworts. People are already know about mosses, but for non-plant people, I'm sorry that I have to say this, but liverworts are special and cool because they have two growth forms. We have leafy liverworts and thyroid liverworts. And in this study, we will focus on the leafy liverworts. So when we talk about trait and function, we usually describe them in the context of adaptations. Organisms, for example, plants, evolve particular traits in response to different habitats. For example, cactus that they modify their leaf structure when they inhabit a dry environment, as well as the flowering plants that are changing their color when they are inhabiting different soil types. So the important parts of plants is the branches. So the evolution of branching is very important to the diversification of plant architecture. We see that many studies about branching patterns in vascular plants in terms of evolution or physiological process. However, in non-vascular plants, there are still limited studies about the branching patterns, especially the study that is supported by the molecular data. So branching types in leafy liverworts, just because leafy liverworts are very, very small, we cannot see them directly by our eyes. We only see them in under the microscope. So we divided the branching in leafy liverworts as a terminal branching and intercalary branching. We divided them, that terminal branching that close to the apex without specific structure under and the base. While intercalary branchings arise from lateral and ventral side with the specific structure on their base called the collar. So this study is mainly inspired by Rudolf Schuster, the expert of liverworts and hornworts, and he dedicated his life to study about the morphological and also the systematic of this taxa. And one of the interesting study one of these his studies is that about the branching evolution in leafy liverworts. So he hypothesized that branching types in leafy liverworts have strong phylogenetic signals. That means closely related taxa tend to have similar branching types more than expected by chance. The other interesting hypothesis from him was the ancestral state in leafy liverworts was branching plasticity, with a combination of multiple types within the taxon. Then over the evolutionary times has become more fixed. However, these hypotheses were based on qualitative data. There is no quantitative data that support this hypothesis. Using the modern techniques recently, so I have the questions about my research. Do branching types really have phylogenetic signals across taxa? Because this is important in systematics when we use the branching data to divide or to delimit the species. My second question is, do ancestral taxa really exhibit more complex combination compared to derived taxa? And my third question, so if there's a evolution in branching, does the environment contribute to this process? Does the complexity of branching correlate with the environmental factors? So to answer my questions, I use study system liverworts in Lepidoziaceae. It has around 1,800 species with remarkable morphological diversity and with the diverse branching types, and they also inhabit in various habitats. And this talk will be divided into three parts. First, we will talk about the backbone phylogeny of Lepidoziaceae. The second one, we will talk about the phylogenetic signals and the branching evolution. And the last part, we will discuss about the phylogenetic comparative analysis. For the first part, for backbone phylogeny of Lepidoziaceae, we gather data from GenBank using custom R code to define the gene regions that cover as many taxa as we can, and then we build the maximum likelihood tree using IQ3. The result shows that these 4 chloroplast regions with 146 species support the circumscriptions proposed by Cooper and Reyes et al. This is we are pretty happy because using our threshold, we support the classification of Lepidoziaceae. And for the phylogenetic signals and branching evolution, we gather data from the literature and online portal, as well as the herbarium specimens from University of Tennessee Herbarium. We identified the branching types in Lepidoziaceae, and we identified 5 branching types within this family. Not only the branching types, we also observed that one species could exhibit more than one type. So we count them as the branching combination. We identified 13 combinations ranging from 1 to 4 types per species. Then we use the 5 branching types as a binary data, and then we analyze for phylogenetic signals using these statistics in a K-per package in R. And the phylogenetic signals here, like we depend on the D value. So the D value here, that 4 types have negative D values, that suggesting closely related species retain similar branching types. And the other result is that Bazania type has a moderate D value that suggests more frequent transition in Bazania type along the tree than expected. Then we use 13 branching combination data to build the custom models for the transition rates, and we build the best model for the stochastic character mapping using a Quorum package in R. So for model of branching evolution, we use 13 observed states, and we constrain branching combination as a stepwise process. And we run 3 different models from this matrix using equal rates, symmetrical, and all rates different. And we ended up with the symmetrical model is the best model for the further analysis. So this is the stochastic character mapping that we have for branching evolution. We see that the ancestral type of branching in Lepidoziaceae is Frulania type and Bazania type. And we can see here, like they gain new types more than 100 million years ago as the evolutionary happens. And if we see from the ancestral type with the derived type, we see that multiple gains and losses of branching type throughout the time within the phylogeny of Lepidoziaceae. This suggests that the ancestral step is likely not the complex combination. So then we jump into the phylogenetic comparative analysis. We also use the 13 combination. We extract them as a simple to complex. Like when they are one type, we score them as 1. And then the more complex, when they have 4 combination or 4 types, we score them as 4. And then we also incorporate the data of habitat moisture from the literature. And then we convert this description into the Ellenberg value for habitat moisture. And then we divided them into 3 different values using the minimum moisture, that is the minimum habitat that a species can inhabit, and the maximum moisture as the maximum habitat that they can inhabit. And we also count the average moisture. We also gather data about the habitat type and substrate. For substrate, we define as epiphytic versus terrestrial, that epiphytic, the species can live in the living trees. For generalists and spatialists, we define them as that how many substrate they can inhabit, because we found that one species could only inhabit one substrate, but the other one, they inhabit like up to 9 substrate. So then we run 10 different models using PGLS and PNCOVA in a Caper package in R. And we run 10 different models from very simple to complex model. And we ended up with the complex model with the complexity as a function of minimum moisture level and habitat type as well as substrate type. And we also see the interaction between them. So this plot shows that in x-axis you see that the minimum habitat moisture level, and then the y-axis is the complexity of the branch. So in generally species, both in epiphytic and terrestrial, they tend, as the habitat moisture increase, they tend to reduce the branching complexity. However, this result is not significant. In epiphytic spatial species, they tend to decrease or reduce the branching complexity as the habitat moisture increase. In contrast, in the terrestrial spatialists, they tend to increase the complexity as the habitat moisture increase. This suggests that environmental factors, especially the specific environmental factors or microhabitat, could potentially contribute to the evolution of the branching complexity. In summary, our analysis strongly support the phylogenetic signal hypothesis. That means similar branching types occur more frequently within closely related species than expected by chance. And then the second one, the ancestral states is likely not a complex pattern. And the branching patterns has undergone multiple gains and losses, at least in the family of Lepidosaceae. And then it is environmental factors that likely contribute to the evolution of branching complexity, and adaptive response to the environmental factors, potentially shaping the phenotypic evolution of leafy liverworts. I would like to take a moment to thank everyone in the team, especially my committee members, as well as the funding resources that support this project. And this is the end of my presentation. I will take any questions. Thank you.
Any questions? Okay, so I'll ask one question. So based on do they have any plasticity of the branching patterns, or do you know if you grow them in a higher moisture environment, do they prone to grow more complex version of the branches, or it doesn't? It's pretty invariable inside a species.
Yeah, so we see this in this study. I see that there are imbalanced number of the samples since we just gather from the references. But from our analysis, this is very surprising that the epiphytic ones that tend to reduce compared to the terrestrial. We assume that microhabitat plays important role, especially in bryophytes, for this morphological phenotype.
Okay, thank you. And okay, I think the next speaker can start to put on the slide and we can switch to our last speaker. Well,